Thank you very much, Brenda, and welcome back to everybody. So just to recap, yesterday we spent a lot of time covering clinical trials in some detail. We spoke about the design and analysis of clinical trials, how they support the safety and the efficacy of medical products, and we discussed their importance in special or specific populations. Today, we're going to change the emphasis a little bit, and our first session is focused on the early clinical or non-clinical studies that are done to support the use of an investigational product. These are probably studies that clinicians are less familiar with, generally reported in the investigator's brochure, which clinicians are expected to read. So I think you will find this quite informative. We're going to begin with a discussion on chemistry and manufacturing controls, which you will learn a little bit about the actual properties of the investigational product. And that lecture will be given by Paresma Patel. We will be moving on to a lecture on pharmacology and toxicology in the investigator's brochure. And that lecture will deal with some of the animal studies that support the use of medical products in a trial and some of the other non-clinical studies. And finally, we will turn to the clinical pharmacology. This lecture will be given by Shirley Xiao, and she will be able to tell us all about how the body processes the investigational product. So without further ado, let me hand it over to Paresma Patel to tell us about CMC. Thank you.
Good morning, and thank you for joining me. My name is Dr. Paresma Patel, and I'm a Division Director in CDER's Office of Pharmaceutical Quality. Today, I'll be giving an agency perspective on chemistry, manufacturing, and controls, regulatory considerations, and resources.
By the end of this presentation today, you should be able to understand the regulatory definitions and requirements for drug substances and drug products, describe chemistry, manufacturing, and controls information for IND submissions, and name some potential CMC safety concerns for INDs.
Today, I'll start off with a brief definition of pharmaceutical quality. I'll discuss how chemistry, manufacturing, and controls, or CMC, changes through clinical development. I'll provide some key regulatory definitions. I'll touch on some specific considerations for drug substances and drug products. And I'll end with important guidance documents and resources. I'd like to start with an overview of the mission and vision of CDER's Office of Pharmaceutical Quality. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. One challenge to the manufacturing aspects of an application is that it covers the entire life cycle of the product and is often rapidly changing from an IND stage of development to commercialization. During preclinical stages of development to the point of IND submission, there is typically limited CMC information. As a product's development proceeds through the phases of clinical development to pivotal clinical studies, significant CMC development is happening. CMC development has to be at a stage to assure consistent manufacturability to support a marketing application. Following approval, the CMC information continues to change as suppliers or manufacturers may change, and the manufacturing processes may continue to be optimized. Throughout this process, the CMC team is meeting with sponsors to advise on key aspects of CMC development as they move closer to commercialization. This includes early-stage interactions such as Interact and pre-IND meetings. The CMC team is involved for Type B meetings after each phase of development, and CMC-focused meetings can happen throughout clinical development. Most CMC-focused meetings typically happen around the end of Phase 2. I'll provide a couple of key regulatory definitions that can be found in the Code of Federal Regulations. Shown here is the definition of drug substance, and to summarize, the drug substance is the active ingredient that provides the pharmacological activity or direct effect for the diagnosis, cure, mitigation, treatment, or prevention of a disease. This is essentially the active component of the drug product that affects the human body. The drug product is the finished dosage form, and it contains the drug substance, and it may or may not contain other ingredients. CMC information required for an IND submission are outlined in the Code of Federal Regulations. The main text of the CFR can be broken down into a few requirements for CMC. They include description, composition, and controls of the drug substance and drug product, information on the manufacturer, the manufacturing process, and data to support stability, information to assure identity, quality, purity, and strength of the investigational drug. The regulations specifically emphasize the identification and controls of the new drug substance and raw materials for an initial Phase 1 submission. Guidance documents are routinely published on FDA's websites. These documents provide FDA's current thinking on a topic and outline information that should be provided in IND and NDA submissions. Guidance documents clarify the type, extent, and reporting of CMC information to ensure quality of the proposed clinical studies. These documents help to outline the data needed for regulatory development and are only recommendations. If sponsors veer away from this advice, they should provide justification and scientific reasoning and meet with FDA if needed.
I would like to also take a minute to highlight that the IND assessment teams are multidisciplinary in nature. The teams include members such as clinical reviewers, clinical pharmacology, statistics, pharmacology, toxicology, in addition to potential consults to other centers and other disciplines as needed. During the IND review for an original IND or first-in-human Phase 1 studies, there typically may only be 1 to 2 CMC or product quality reviewers on this team, depending on the specific product being reviewed. However, as development proceeds beyond those early stages of development, the product quality team expands to include sub-discipline expertise. So by the time a marketing application is submitted, the product quality team includes members from the following disciplines: drug substance, drug product, biopharmaceutics, which really considers in vitro dissolution of the final dosage form, drug product manufacturing, evaluation of facilities, microbiology, and finally surveillance and inspections that may be needed for the facilities that manufacture the drug substance or the drug product.
I'll start off focusing on the drug substance or active pharmaceutical ingredient information that FDA generally expects as part of an IND application. First is general information. This contains information about the structure, molecular weight, formula, and the source of the drug substance. The manufacturing process section includes a description of the chemistry for how a drug substance is made and includes both a flow diagram and a written description for each step of the manufacturing process. The characterization data section includes both structural characterization data and characterization of physical-chemical properties. The impurities section provides available information on observed and potential impurities. Sponsors should specifically address differences in impurities between toxicology and proposed clinical batches. The controls part of CMC defines a release specification, which is a predetermined set of criteria used to release batches of drug substance for use. This section includes information on the batches intended for use in the clinical study and those that were used in the toxicology study. And finally, data to support stability for a drug substance should be provided in the stability section. Preliminary stability data is typically sufficient for an initial IND submission.
One key aspect of our regulations is that they are broadly applicable to APIs with significantly variable structures. As part of regulatory submissions, information on the source of drug substance is submitted, and there are a wide variety of APIs that are regulated as drugs that we review in our office. There are drugs manufactured using chemical synthesis, which includes small molecules. Shown here are just a couple of examples: elaxacaftor and nirmatrelvir. Additionally, peptides can also be manufactured using chemical means, such as solid-phase peptide synthesis. Shown here is just an example of plecanatide. And then oligonucleotides, such as siRNAs or antisense oligonucleotides, also fall under this category and can be manufactured using chemical synthesis. Shown here are just the two examples: patacerin sodium as well as tofersen. There are also those drugs derived from fermentation products, such as erythromycin, or semi-synthetic drugs such as paclitaxel, where the portion in red is isolated from the U tree and chemical synthesis is used to modify the structure further. There are a wide variety of drugs that are isolated from natural sources, including drugs from inorganic sources, botanical sources like the synecatechins, and animal sources such as heparin, which is derived exclusively from the intestinal mucosa of pigs. We also review combination products such as antibody-drug conjugates. These are reviewed in collaboration with biologics reviewers with a focus on the chemistry and conjugation of the drug linker or the payload linker. Hopefully the last couple of slides gave you an idea of the molecular variability and science that chemists within FDA have to understand in order to review and regulate products. In addition to the information related to the actual structure and source of the drug substance, the bulk of the review for drug substance is really the chemistry for how the drug substance is made. I'll focus on new molecular entities today because the manufacturing process is typically at an early stage of development when an IND is submitted. In order for us to assess the safety of the drug substance to be used in clinical studies, we ask for a brief description of the synthesis used to manufacture the clinical trial material. This information should be provided in both a written format as well as a synthetic scheme that provides us with a complete picture of all the chemical reagents, solvents, catalysts that are used as part of the manufacturing process, and information on any of the controls for the purity and quality of these materials. In addition, as part of the manufacturing section, we often get information on any preliminary and process controls, which are really the analytical test to ensure reaction completion as part of this at each step of a process. For our first in-human NME IND, the emphasis really is on the difference in the quality of toxicology batches that were used for non-clinical studies and the clinical batches planned to be used. And we work closely with our pharmacology and toxicology colleagues to assess any differences between these manufacturing processes or the quality of these batches. I would also like to just emphasize that FDA does not expect that all of this information is fully understood at the time of IND submission. We're aware that the manufacturing process is being optimized for scale, yields, purity, and to minimize use of toxic or dangerous chemicals. Additionally, we're also aware that minimal steps are usually conducted under GMP at these early stages of development, and the commercial GMP manufacturing process has not been developed or finalized quite yet. FDA does encourage that sponsors initiate a CMC-focused meeting prior to initiation of pivotal clinical studies to really address any CMC challenges or to come to agreement on any specific CMC aspects related to manufacturing of an API. Next, I'm going to move on to talk about the characterization that's expected for a drug substance. I'll group this into two categories: structural characterization data and physical-chemical characterization data. First is the structural characterization data. This is the data to support the proposed structure of the drug substance itself. And we recognize that structural characterization of the drug substance may be limited early in development, but as part of this characterization, we need enough data to assure identity, and we want to see submission of multiple characterization techniques to support the structure of the drug substance. Some of the standard techniques for small molecules include spectroscopic data such as NMR, UV-Vis, or IR. I would also like to note that for complex drug substances, we typically expect additional characterization. For example, for a peptide or oligonucleotide, we would expect characterization to confirm the sequence. As part of the drug substance characterization data, we expect submission of the raw spectral data itself. In addition to the raw data, we expect interpretation of this data as well. For example, assignment of peaks for an NMR or functional group assignments for an IR. During review of the IND itself, the FDA reviewers will then evaluate the totality of the characterization data and the provided interpretation, and will assess that information independently and provide any comments or developmental phase-appropriate advice moving forward. We acknowledge that identification of some peaks may be ambiguous early in development, as sponsors may be in the process of identifying impurities that are present at low levels. I want to briefly describe some of the physical-chemical characterization data that should be considered in the context of its criticality to the final drug product and drug product formulation. These types of attributes include physical attributes, including appearance and physical form of the drug substance. For example, is the drug substance a solid or an oil, and what color is it? We would want information on solubility in aqueous solutions, as well as any of the organic solvents that may be used as part of the manufacturing process. Information on particle size distribution may be limited at an initial IND submission, but the potential impact of this attribute for the drug product should be considered as additional clinical batches are manufactured. Information on polymorphic forms, as well as hygroscopicity, or the propensity of the drug substance to absorb water. Information on whether or not the drug substance is hygroscopic should be provided, and an assessment of the implications for the final drug product dosage form should also be considered. We generally encourage that sponsors monitor and characterize these attributes through development and assess the criticality of these physical and physical-chemical attributes to the final drug product, with an understanding that there may be changes to the drug product in early development. As part of the characterization section of the drug substance, there's typically an assessment of impurities. Generally, an impurity is any component of the drug substance that isn't the drug substance itself, and we typically want to minimize the presence of impurities in drug substances. Shown here is one relatively simple step of a drug substance manufacturing process to illustrate potential sources of impurities. Shown here is the reduction of a nitro group to an aniline using palladium catalyzed hydrogenation in the presence of hydrogen gas and ethanol as a solvent. From this one relatively simple step, you can see that there are multiple potential sources of impurities, including a heavy metal or elemental impurity, such as residual palladium. Residual ethanol could be a residual solvent that could carry forward to the drug substance. Any unreacted starting material would be considered a potential impurity, and in this case, the starting material has a structural alert, which is a functional group that may be of concern from a genotoxic perspective. In this case, the product of the reaction is also has a structural alert and could be a genotoxic concern. As a drug substance reviewer, we're essentially tasked with looking at the controls for the starting materials, intermediates, and final drug substance to assure that all of these potential impurities are adequately controlled and they don't pose any safety risk to patients. At early stages of development, we really want to know what control strategies are available for observed and potential impurities. And while we don't expect that all of these impurities are necessarily identified as part of the initial IND submission, we do want to understand the proposed strategy to further identify and control these impurities. And again, the emphasis in a phase one person-human study is really to address any differences in impurities and impurity profiles between the toxicology and clinical batches.
Now I'll move on to the overall controls for release of the drug substance and drug product. The specification is the predetermined set of criteria used to test and release batches for clinical use. The drug substance specification will be unique compared to the drug product specification, but the overall format and presentation is similar. The specification table provides the list of test methods and proposed limits to assure that clinical batches of drug substance and drug product meet the regulatory requirements, assuring identity, strength, quality, and purity. In the initial IND, the analytical test method can be briefly described or referenced to a compendial test. The limits for an IND are typically flexible based on the available toxicology and clinical batch data, and are often further defined and narrowed as development proceeds and nears commercialization. Typically, we would recommend that any critical attributes for the product be monitored as a test within the specification. For example, if the particle size of the drug substance is important to the drug product manufacturing, we would recommend this attribute be monitored through clinical development. As part of the IND submission, we expect to see either tabular batch data or certificates of analysis for the proposed clinical batches, tested according to the proposed specification. Shown here is a representative drug substance specification. Specifications include three main elements: the test attribute, the analytical method, and acceptance criteria. We recommend that you capture your rationale in a thorough justification of specification document. FDA generally is more flexible with acceptance criteria for early stages of development, with the understanding that there's very limited batch data available. What we're looking for is that there is sufficient control of the identity, purity, impurities, and quality of the drug substance to assure safety. A better understanding of the drug substance manufacturing and quality is expected as development proceeds, and we would expect phase-appropriate updates to the specification table as additional process and product understanding is gathered and the development program moves closer to commercialization.
I would like to briefly discuss stability data for drug substances. Stability studies are conducted to measure how the quality of a drug varies with time under the influence of environmental factors. And these studies are important to establish a useful retest period or shelf life for a drug substance when stored under long-term storage conditions. Stability specifications may be different from release and should include attributes that are likely to change during storage. For example, attributes such as appearance, purity, water, content, or polymorphism are typically considered stability indicating attributes. Long-term storage testing is conducted under the recommended storage conditions for the shelf life or retest period of the drug substance and is used for labeling. Accelerated testing is typically run under conditions with increased rates of degradation. The stability section of an IND should clarify the tests that are run to monitor stability. Preliminary data on representative material for both drug substance and drug product is typically acceptable for an initial IND submission. If stability information on the clinical batch data is available, that information should also be submitted in the IND. If stability data is not available, we ask that sponsors provide a commitment to continue testing the clinical trial material through the duration of the proposed clinical study.
I would like to now touch on specific information that is needed for a drug product compared to a drug substance as part of an IND submission. For the drug product, it's important that we have a description of the dosage form and any information to justify the safety of a novel technology or any complex drug product formulation. It's particularly important to clarify any patient administration information for the proposed dosage form. For example, a drug product that is administered parenterally, it'd be important to consider compatibility issues with any administration devices or solutions that may be used to dilute the drug product prior to administration. The quantitative composition of the drug product should be provided, and this includes a list of the inactive ingredients or excipients. We ask for information on the compendial status or quality of those excipients. If novel excipients are used, additional CMC information may be needed to assure quality of those excipients. Additional information may also be needed to assure safety of any animal-derived excipients. If using novel excipients, we generally recommend discussing your approach during a pre-IND meeting to assure adequate data is provided in the IND submission. The drug product manufacturing process should be described with both a brief written description and a flow diagram. And additional information on the sterilization process should be provided if it's applicable to your drug product. Similar to the drug substance, the control of drug product is essentially a specification that includes a set of criteria to demonstrate quality of the drug product prior to releasing batches. This section includes information on impurities that are specific to the drug product, as well as the proposed batch data for use in the intended clinical study. Finally, information on how the drug product is packaged and preliminary stability data should be provided in the IND submission.
There are a few miscellaneous requirements that are outlined in the IND regulations. One is the requirement for a product label that includes a caution statement, which is shown here. Additionally, the submission of an investigator's brochure that has a brief description of the drug substance and the drug product formulation should be provided. Additionally, the requirement for submission of an environmental assessment or a claim for categorical exclusion are also referenced here. I want to point out that INDs generally are categorically excluded from the requirement to submit the environmental analysis, but we do need that claim and request for that claim in the IND submission. Similar to the drug substance specification, the drug product specification is the predetermined set of criteria to ensure identity, strength, quality, purity, potency, and bioavailability of the drug product. The specification includes the attribute being tested, the analytical procedure, and acceptance criteria. Shown here is a representative drug product specification that includes some key tests, including identity and appearance, assay and impurities, content uniformity, and water content. In this case, we have controls for reconstitution, particulate matter, osmolality, sterility, and endotoxins, which are dosage form specific. I would like to briefly touch on the information that should be provided on the container closure for a drug product and information to demonstrate stability. For an IND, a brief description of the container closure and packaging components can be provided with any precautions that may be needed to ensure protection of the investigational drug during clinical studies. Similar to the drug substance, the drug product stability data is required to establish the shelf life and set an expiration dating period for the final product. Drug product attributes that are likely to change and impact the quality or performance of the drug product should be investigated on stability testing. Stability data should be conducted under long-term storage conditions and accelerated conditions. Preliminary data on representative material for drug product is acceptable for an original or initial IND submission. If stability information on the clinical batch is available, that information should also be provided in the IND. Additionally, in-use compatibility data should be provided to demonstrate compatibility of the drug product during administration. These studies should include any reconstitution steps and data to support hold times.
Some of the CMC issues that are likely to lead to clinical holds of ongoing studies include the following. Questions related to adulteration or purity of the materials used to manufacture the drug substance or the drug product. If the impurity profiles are not sufficiently characterized or the safety of the impurities is not justified by supportive non-clinical studies. Any impurities that are known to be highly toxic or pose potential risks to patients. Unreliable analytical methods that may undermine the confidence in the data provided in the IND. Insufficient batch data to demonstrate manufacturability of either the drug substance or drug product. Any significant stability issues. Lack of sterility assurance or endotoxin controls could pose significant safety issues or risks to patients. Challenges with the drug product formulation, such as formation of particulate matter upon storage of a parenteral drug. And finally, issues related to cGMPs at the manufacturing facilities. One way to avoid issues at filing of an IND includes consistent communication with the FDA through industry meetings and paying close attention to the advice given during those meetings. Prior to an IND submission, every sponsor is granted at least one pre-IND meeting. As part of this meeting, the sponsor of the IND submits a brief package that describes the background of the product and proposed clinical development. CMC-specific focus areas during a pre-IND meeting include the following: whether the manufacturing process and characterization data appear reasonable, the acceptability of the drug substance and drug product controls or specifications, the overall stability data and stability design, whether the impurity levels and controls appear reasonable for phase 1 studies, adequacy of the toxicology batches to support use of the proposed clinical batches, and any overall potential gaps or hold issues that the CMC team may find. I would like to end with a few resources, including important websites as resources, IND-focused guidance documents with CMC advice, and resources for expedited programs. This slide provides resources that are specific to control strategies for impurities that could be used during development.
And finally, I have 2 challenge questions for today's presentation. Challenge question number 1. The regulatory term for an active ingredient intended to furnish the pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or any function of the human body is A, drug product, B, impurity, C, drug substance, D, intermediate. The correct answer is C, drug substance. Challenge question number 2. The following are CMC safety concerns, except for which one? A, impurities of known or potentially high toxicity. B, insufficient batch data. C, lack of sterility assurance or endotoxin control. Or D, the manufacturing process route for the marketing application has not been finalized. The answer is D. All of the other options are potential CMC safety concerns. However, the manufacturing process for marketing is typically not finalized until closer to conducting the pivotal clinical studies.
That concludes my presentation, and I will be back later to respond to some of your questions. But now I will turn it over to Nikolett Biel to share her insights on pharmacology, toxicology in the Investigator's brochure.
Thank you to Dr. Patel's insightful presentation on CMC. We are now going to switch gears and talk about the pharmacology and toxicology information that you will find in the Investigator's brochure, or the IB for short. As Leonard mentioned, I am Nikolett Biel. I am a non-clinical reviewer in the Division of Hematology, Oncology, Toxicology, supporting the Office of Oncology Drugs at the FDA. The learning objectives for this presentation are to be able to describe the types of non-clinical information provided in the IB and understand the relevance of this information to drug development. Non-clinical information includes pharmacology, safety pharmacology, pharmacokinetics and ADME. I will later touch on the meaning of ADME for clarification. Toxicology, which includes general toxicology, genetic toxicology, and other toxicology studies, including reproductive toxicology. Additionally, throughout the presentation, I will refer to guidance documents such as International Council for Harmonization, or ICH, and or FDA guidances for industry that are available for drug developers to aid in the types and timing of non-clinical studies needed to support drug development. I will point to these references throughout the presentation and have also included a reference slide at the end of the presentation. In the non-clinical section of the IB, you will find information on the pharmacology of the drug. Let's take a closer look at what this information provides. The pharmacology section of the IB will provide data including proof of concept, or POC studies, demonstrating the mechanism of action of the drug, as well as its activity in vitro, for example, in cells, and or in vivo, for example, in animals, such as anti-tumor activity in tumor-bearing mice for oncology drug products. The pharmacology studies will also demonstrate the primary pharmacology, or data on the intended target, and secondary pharmacology, or data demonstrating activity, or preferably lack of activity, on unintended or off targets. Furthermore, especially for biological drug products, the pharmacology studies can support the selection of species for toxicology studies. For example, some antibody drugs only bind human and monkey drug targets and may not bind the rodent target. In that case, the monkey will be considered the only pharmacologically relevant species to assess the safety of the drug. The amount of non-clinical data provided may vary depending on the type of drug product, whether a small molecule or a biologic. Biologic pharmaceuticals, such as immune-modulating antibodies, may have more pharmacology data, as animals may underpredict toxicities due to differences in sensitivity between humans and animals, or there may not be a pharmacologically relevant species for toxicology testing. So the safe starting clinical doses generally rely on many of the pharmacological data to determine what is called a minimally anticipated biological effect level, or MABEL. The amount of data may also vary depending on the indication. For example, pharmaceuticals intended for oncology or non-oncology indications. Additionally, the stage of drug development will also dictate the amount of data provided by the sponsor. Most pharmacology data will be submitted at the first-in-human stage, and additional data may be submitted to support different indications or combination regimens that may be evaluated during clinical drug development. Following the pharmacology section, you will find information on the safety pharmacology of the drug. Safety pharmacology studies are conducted to assess the potential effects of a drug on physiological functions on vital organ systems, including the potential for adverse effects on the heart, central nervous system, and respiratory system. Assessments on the cardiovascular system generally include in vitro assessments on the human ERG potassium channel and in vivo assessments mainly in non-rodents, for example, dogs or monkeys, including blood pressure and electrocardiogram measurements. Together, these data could identify a potential risk to normal heart functions. Central nervous system assessments include motor activity, behavioral changes, coordination, sensory or motor reflex responses, and body temperature. The respiratory system assessments include the measurement of respiratory rate. Central nervous system and respiratory system assessments are commonly conducted in rodents. Safety pharmacology studies can either be conducted as standalone studies, usually a single dose, or as part of the repeat dose toxicology studies. Both will use the intended clinical route of administration. Information on the conduct, timing, and exceptions for these studies can be found in the ICH S7 guidance for industry. Following the safety pharmacology information, you will find information on pharmacokinetics, or PK, and ADME, which means absorption, distribution, metabolism, and excretion. Pharmacokinetics is the drug's movement through the body as it passes through 4 different phases known as ADME. These studies describe how a drug moves throughout and is processed by the body. PK describes parameters such as maximal exposure, steady state exposure, the half-life of the drug, and clearance of the drug. These factors depend on ADME. Absorption is the process from administration, for example, taking a tablet, to systemic exposure or presence of the drug in blood plasma. The absorption can affect the speed and concentration of the drug in the systemic circulation. Distribution is the presence of drug in different tissues that may affect the efficacy or effectiveness and toxicity of the drug. Metabolism describes the different ways a drug may be broken down and identifies potentially human-specific metabolites, which would not be present in toxicology species. Excretion describes the mode of elimination of a drug, for example, through urine, feces, or bile. Toxicokinetics, or TK, is the study of ADME measured in a toxicology study with doses that may or may not be toxic to animals. TK informs on parameters such as dose proportionality and exposure. The exposure to some drugs is proportional to the increase in dose. For example, an increase by thirds in the dose would be an increase by thirds in the exposure. However, the exposure to other drugs may be less than or more than dose proportional. At times, exposure to drugs may reach a saturation point where increase in dose does not correlate to increase in exposure. TK can also inform on whether there may be differences in exposure between male and female animals, which can influence the determination of a safe clinical starting dose. TK also informs on the potential for accumulation of a drug or increased exposure to a drug upon repeat dosing. In some instances, for example with biological pharmaceuticals, repeated exposure to the drug may lead to loss of systemic exposure due to the formation of antidrug antibodies or ADAs that lead to faster clearance of the drug. Following the PK and ADME information, you will find information on the toxicology assessment of the drug. General toxicology studies prior to administering a drug to humans are conducted to determine whether the proposed clinical study is reasonably safe. Dr. Shabnam has touched on this during yesterday's seminar and how we use doses from the toxicology studies and animal to human scaling to determine a safe clinical start dose. The toxicology studies help determine a reasonably safe clinical start dose and guide dose escalation. They can identify potential dose-limiting toxicities that help inform the clinical safety monitoring and may assess potential toxicities that cannot be identified in the clinical trials. For example, potential acute histopathological changes in tissues and organs due to drug exposure. The toxicology studies use the same route of administration and schedule in the animals and intended in humans in the clinical study. Studies are generally conducted in two species, a rodent and non-rodent. The ICH M3R2 guidance describes the duration of clinical studies relative to clinical development of pharmaceuticals. And ICH S9 and S9 questions and answers is followed for oncology pharmaceuticals. For non-oncology drugs, the duration of the toxicology studies should cover the duration of the clinical trial. Studies of 2-week duration in 2 species support a clinical trial lasting up to 2 weeks. Toxicology studies supporting a clinical trial lasting between 2 weeks and 6 months should follow the same time frame as the intended clinical study. Studies in rodents of 6-month duration and in non-rodents of 9-month duration will support a clinical trial lasting longer than 6 months. For oncology pharmaceuticals to support a first-in-human trial, a study of 1-month duration in 2 species would support a trial, and additional studies of 3-month duration in 2 species are generally warranted to support a registrational trial intended to support a marketing application. For biological pharmaceuticals, there may only be 1 species for toxicology studies or none. I will discuss this in more detail on the next slide. Acceptable safety margins from toxicology studies to the clinical start dose depends on the indication of the drug, whether it's for an oncology or a non-oncology indication. The guidance estimating the maximum safe starting dose in initial clinical trials for therapeutics in adult healthy volunteers for non-oncology and ICH-S9 for oncology drugs can be referenced. Sponsors may use a variety of species to conduct the general toxicology studies. The commonly used species for rodent include mice and rats, and for non-rodent, the dog, monkey, and rabbit. Species selection may depend on the type of pharmaceutical, whether a small molecule or biologic. For small molecule drugs, rats and dogs are commonly used, and species selection may be based on comparable drug metabolism and types of metabolites present compared to humans. For biologics, species selection will depend on the target binding. Occasionally, only the monkey is a relevant pharmacological species because the antibody may not bind the rodent or other non-rodent drug targets. This is often due to minimal or lack of sequence homology of the drug target between human and rodent. In that case, toxicology studies are often conducted in the only pharmacologically relevant species. There are cases, although not common, where none of the animal species are pharmacologically relevant. In that case, a safe clinical start dose is determined based on the pharmacology studies. The next 4 slides include real examples where toxicities observed in animal repeat dose toxicology studies informed clinical monitoring. These examples are all from oncology, since I am a non-clinical reviewer in the oncology division. I do want to note that for oncology pharmaceuticals, we allow some toxicity in the animals to support a clinical dose in patients with advanced cancer, compared to animal studies supporting a non-oncology indication. One example is a microtubule inhibitor drug developed for the treatment of advanced solid tumors. The drug led to irreversible optic nerve degeneration at the mid and high doses in the rat repeat dose toxicology study. Not only did this finding limit the dose from which the safe clinical start dose was calculated from, but also due to concerns the clinical monitoring was increased, including optic exams and imaging, and the information was added to the clinical protocol and patient-informed consent form. The second example is epigenetic targeting drugs that are being developed for the treatment of solid tumors and hematologic malignancies. The drugs led to malignancies or lymphoma in rat 3-month repeat dose toxicology studies. The secondary malignancy has also been observed in clinical trials. Based on concerns, the patient population being studied were considered, meaning the risk benefits were assessed and indications with indolent or less refractory tumors may have been removed from the trial. The information was also added to the IB and the patient-informed consent form. This information is also found in the warnings and precautions of the label of an FDA-approved drug in this pharmacological class. The third example is an antibody-drug conjugate, or ADC, with a topoisomerase I inhibitor. This ADC led to severe kidney toxicity, including increased urine protein, proteinaceous casts, and multifocal necrosis in the monkey repeat dose toxicology study. At the time of the IND review, due to the kidney toxicity in monkeys, the sponsor was asked to lower the clinical start dose and modify the dose escalation. Kidney toxicity was observed in patients during the dose escalation, which led to additional clinical safety monitoring, an update to the IB, and the patient-informed consent form to inform investigators and patients of the risk for kidney toxicity. In the fourth and final example, an immune agonist was being developed for the treatment of patients with urothelial carcinoma to be dosed locally in the bladder. This route of administration should localize the drug within the bladder and have minimal to no systemic exposure. In the monkey repeat dose toxicology study, a low level of systemic exposure was observed when monkeys were dosed locally in the bladder. The clinical team was informed that if systemic exposure is observed in humans, then there may be a risk for cytokine release syndrome or CRS with this drug. The clinical safety monitoring was updated to add up to 6 hours of monitoring of patients following the first dose in the event that systemic exposure may lead to CRS. Following the general toxicology studies, you will find information on genetic toxicology. Some drugs may be carcinogenic or have the ability to cause cancer by inducing tumors, increasing their incidence, or shortening the time it takes for tumors to occur. Results from carcinogenicity studies are generally not available until the time of product approval. To note, carcinogenicity studies are not warranted for pharmaceuticals administered infrequently or for a short duration of exposure, such as anesthetics and radio-labeled imaging agents per ICH S1, and for advanced cancer indications, but may be needed for early cancer indications per ICH S9. In lieu of carcinogenicity studies, genetic toxicology assessments are used as a surrogate and help address concerns about the risk to humans during clinical trials. Additionally, results of genetic toxicology studies can inform on the duration of contraception. Genetic toxicology studies are generally conducted with small molecule drugs. A general battery of genetic toxicology studies includes 2 in vitro assays, one to test for gene mutation in bacteria, and another to detect chromosomal damage in mammalian cells. An in vivo assay commonly conducted in rodents to detect chromosomal damage to hematopoietic cells. Other genetic toxicology assays may be conducted. Further information can be found in the ICH S2 guidance. Relative to clinical development, the bacterial gene mutation assay is warranted to support a single-dose clinical trial. Additionally, the in vitro chromosomal damage assay is needed if proposing multiple clinical doses. The complete battery of studies, including the in vivo assay, should be submitted prior to a phase 2 trial. To note, for oncology pharmaceuticals, the genetic toxicology studies may be submitted at the time of the marketing application. However, results from genetic toxicology studies are needed to support clinical trials in healthy volunteers, which are typically conducted for food effect or drug-drug interaction assessments for oncology pharmaceuticals. Information on other toxicology studies in the IB include results from reproductive toxicology studies. Some drugs can cause teratogenicity or fetal malformations. One well-known example is thalidomide. This drug is used to treat leprosy and multiple myeloma, but was also prescribed to pregnant women for nausea and insomnia in the 1950s and 60s. The use of thalidomide in pregnant women in Europe, Australia, and Japan led to over 10,000 births with severe limb malformations. It was banned from use in pregnant women in 1961. Studies have shown that the sensitive period to thalidomide during pregnancy was approximately days 20 to 34 post-fertilization. The potency of the drug and the relative short time period between drug exposure and manifestation of side effects allowed the link between the two to be made. Reproductive toxicology studies are now a standard assessment during drug development to prevent side effects such as those seen with thalidomide. The timing of these studies depends on the patient population, whether patients are male or female, with or without childbearing potential. The timing also depends on the type of drug, whether it's a small molecule or a biologic, and the intended indication, whether it's non-oncology or oncology, and expected duration of treatment, whether it's short-term, usually the case with patients with advanced cancer, or long-term, such as years in non-oncology settings. Reproductive toxicity assessments are included in the general toxicology studies to inform on potential effects on fertility. Embryo-fetal pre- and postnatal periods generally follow the M3, S5, and S6 ICH guidelines and discuss the type and timing of these studies during drug development. ICH S9 is followed for oncology drugs, and generally only embryo-fetal toxicology studies may be warranted to support a marketing application. Some of these studies are not available at the time of the first-in-human clinical study. To prevent a potentially embryo-fetal or teratogenic drug to lead to adverse effects on the unborn fetus, clinical studies include testing for pregnancy prior to drug exposure and the use of effective contraceptive methods during treatment and for a period of time after the last dose of drug. In summary, non-clinical studies are an important part of drug development to inform on the mechanism of action of the drug, the safety of the proposed first-in-human clinical dose, the potential toxicities that may be observed during clinical studies, and inform on possible long-term adverse effects during the life cycle of the drug post-approval. Here I listed non-clinical resources and guidances that I have touched on during the presentation. Before wrapping up, I have two challenge questions for you. Challenge question 1. The safety pharmacology core battery does not include A. Cardiovascular system, B. Central nervous system, C. Respiratory system, or D. The renal system. I will give you a few seconds to think about your answer. If you picked answer D, the renal system, you are correct. The core battery of safety pharmacology studies include assessments of the cardiovascular, respiratory, and central nervous systems. Challenge question 2. The complete battery of genotoxicity studies for non-oncology indications should be completed A. Prior to the first-in-human IND stage, B. Prior to initiating a phase 2 study, C. Prior to submitting a registrational clinical trial intended to support drug approval, or D. At the time of the marketing application. I will again give you a few seconds to think about your answer. If you picked B, prior to initiating a phase 2 study, you are correct. If you picked D, at the time of the marketing application, you would have been correct if I singled out oncology pharmaceuticals, given the timelines for these studies vary depending on the intended patient population. That concludes my presentation. Thank you for your attention. I will be back to respond to your questions, but now I will turn it over to Dr. So to share her insights on clinical pharmacology.
Good afternoon, everyone. Thank you for joining. Today, I'll be speaking to you about clinical pharmacology and how we fit into early drug development. Since the overall objective of my talk today is to explain a little bit about clinical pharmacology and discuss its critical role in early drug development, I'll be focusing on two main aspects. First, to understand the scientific underpinnings of clinical pharmacology as a discipline. And then, in the second part of my presentation, I'll be giving an in-depth overview of early clinical studies. There will be a focus on various aspects, such as study conduct, the information gathered as a result of these studies, and how to use that information in our drug labeling. And at the end, I'll also touch on a couple of developing areas in our field, including PBPK and model-informed drug development. Okay, so clinical pharmacology, what exactly is it? Like the old saying goes, if you ask 5 different clinical pharmacologists, you'll get 5 different answers. But in its simplest form, it is the study of the pharmacokinetics, or PK, and pharmacodynamics, or PD, of a drug in humans. PK is what the body does to the drug. It encompasses the processes of absorption, distribution, metabolism, and excretion for most drug products. PD, or pharmacodynamics, is what the drug does to the body. These two concepts are represented by these graphs on the right-hand side, where on the top graph you have a concentration of drug in blood or plasma of some type of biological matrix over time. And in the bottom graph, you see an effect, some sort of biological measured effect over time. So what happens when we put it all together? We get a pretty powerful clinical pharmacology tool called a PK-PD relationship or exposure-response relationship, depending on the measured outcome. When we take a concentration over time profile and combine that with an effect over time profile, we get effect over concentration. So what we can really see is the effect of a drug over varying concentrations of the drug. And so in this way, we're able to predict some sort of quantified biological effect given any concentration of drug in the body. Given these tools at our disposal, how do clinical pharmacologists use them to contribute to the drug development process? Well, we like to say that we own the dose, and by that I mean we help determine the dosing regimen of a drug, how much to give, how often to give it. We even determine if a drug needs to be adjusted due to various intrinsic or extrinsic factors. In other words, are we giving the right drug at the right dose, at the right time, to the right patient? These are all very important questions that are asked of clinical pharmacologists and that we help to address through studies in early drug development. Now, I've mentioned ADME before, and assessing these processes gives us key information on the clinical pharmacology properties of a drug. The A in ADME stands for absorption, and it's simply the process of a drug reaching systemic circulation. It doesn't apply to all routes of administration. For an IV administered drug, there is no absorption since it's delivered straight into a vein and directly into systemic circulation. But absorption through an oral formulation, let's say, can be impacted by several factors in the body, such as eating food or pH-altering medications or other concomitant medications, alcohol, transporters, or drug metabolizing enzymes being altered in the gut. Important information that we typically communicate in the drug labeling, or USPI, in section 12.3 are the bioavailability of a drug, the time to reach maximum concentration, or Tmax, as well as the effect of food. The D in ADME stands for distribution. Distribution is the process of drug being distributed to different areas of the body, so it's just how it sounds. It is described by volume of distribution, which is a numerical descriptor that's given in the units of liters. It tells us the drug's propensity to either remain in blood or distribute to various organs throughout the body. So the higher the number, the more likely the drug is to distribute to various organs. Some factors that we need to consider for distribution are, does the drug bind to plasma proteins? Is the extent of protein binding either concentration or time dependent? These are important questions because only the free or unbound drug is pharmacologically active. Sometimes the measurement of unbound drug is recommended when interpreting data. In addition, is information on the distribution to cerebrospinal fluid relevant for this drug? Important information that we typically communicate in the drug labeling under section 12.3 include the percent protein bound of the drug, as well as the volume of distribution. Now here I've combined the M and the E. They stand for metabolism and excretion, and they fall under the general category of elimination. You may have heard that term before. Elimination is a broad category that includes metabolism and excretion. So metabolism in technical terms is the process by which lipophilic molecules are converted into more hydrophilic ones, so they can be excreted by the body. And excretion is the irreversible loss of drug from the body. So once it's excreted, it is not coming back. Factors to consider under this section is how is a drug metabolized? How is it cleared from the body? This usually involves either hepatic metabolism or a combination of hepatic metabolism and renal excretion. And what fraction of the drug is being metabolized? And are there active or toxic metabolites that are being produced? Important information that we typically communicate in the labeling under section 12.3 include the half-life of a drug, the major route of elimination, and the percent excreted in either urine or feces or both. And here I have a few other important considerations for a drug. Is it a narrow therapeutic index product or drug? If it is, that means that very small or slight changes in drug exposure could significantly impact the drug's safety or efficacy, which then may require therapeutic drug monitoring in clinical trials and in clinical practice in order to minimize these toxicities and also to minimize the loss of efficacy. Also, is this drug a significant inhibitor or inducer of either cytochrome P450 enzymes or transporters? If so, this drug may need to be evaluated in further drug interaction studies. And now I'll move on to the next section of my talk, which is giving a brief description of early clinical studies that involve clinical pharmacology. First, I'd like to say a word or two about the timing of clinical pharmacology studies. We're often asked this question about when should I conduct them? Well, the answer is as early as possible. Right after data is collected from preclinical development and an initial IND is submitted that contains the first-in-human study in the US, we begin the start of clinical development. And during this process in phase 1, phase 2, and phase 3, the earlier you can get information from clinical pharmacology studies, the better, because in that way you can help inform the next study or the next phase that comes through, and then that helps maximize the chances of success for your drug. And early phase studies are usually designed to investigate the safety and tolerability, and sometimes to identify the maximally tolerated dose and the PK of the drug in humans. So with all of this information, this is critical for each step in the drug development process. So I mentioned that the start of an IND is the first-in-human study. So usually a first-in-human study entails a starting dose that's determined by some sort of preclinical tox study. And it includes a randomized placebo-controlled study in healthy volunteers. Sometimes patients are used in these studies, but that's fairly rare. And what we're looking to gather from a study like this is some initial safety and tolerability data, possibly the maximally tolerated dose, general PK characteristics of the drug, maybe it's highly variable, what is the PK linearity, and possibly some steady state parameters from the multiple ascending dose study. And preliminary exploration of drug elimination is also ideal. Next, we have the ADME study. which is recommended for nearly all drugs in development. Its objective is to understand the clearance mechanisms of the drug and any and all metabolites in humans. We just put out a new guidance on mass balance studies, and you can find that in the link here, or you can navigate to the FDA guidance website. It's sometimes referred to as mass balance studies, but a lot of folks also call it an ADME study. What it involves is you getting a radio-labeled drug molecule, administering it usually as a single dose to healthy volunteers, and using the intended route of administration, and then in the end, measuring concentrations of the parent and any and all metabolites to determine the relative amount of radioactivity in plasma and other biological matrices such as urine and feces. What we're hoping to learn from this study is to determine the overall pathways of metabolism and excretion for the investigational drug, finding out if there are any metabolites in circulation, and then determine if what the abundance of those metabolites are relative to either the parent drug or total drug-related exposure. Next up, we have bioavailability studies. It's important to conduct these in order to evaluate the rate, or Cmax and Tmax, and extent, or AUC, of absorption of a drug from a test formulation and then becomes available at the site of action. It's typically a crossover single-dose study in healthy subjects where the extent and rate of absorption of the parent drug and any major metabolites, if any, are measured. We can assess either relative bioavailability, which is testing one formulation versus another, or absolute bioavailability, which is the investigational formulation versus the IV formulation of itself. What we hope to learn is the amount of drug that reaches systemic circulation from a tested formulation. And we do have a guidance for industry that was just put out or updated last year on the FDA website. Moving on to my favorite kind of study, a food effect study. Since we all eat food every day, it is important to evaluate the impact of food on a drug's systemic exposure. So the overall objective is to evaluate the effect of food on the rate and extent of drug absorption from a given formulation, and this involves all oral formulations, of course. It involves a single-dose study in healthy volunteers using the highest therapeutic dose of a drug product. In the fed state should be an FDA high-fat, high-calorie meal, although other meals can also be studied. But the FDA recommended high-fat, high-calorie meal is contained in the new food effect guidance, which was just updated last year, and you can find that on the FDA guidance website as well. The PK assessments that come out of a food effects study are very similar to a bioavailability study. No food effect could be concluded if the 90% confidence interval of the fed to fasted ratios for Cmax and AUC fall within the 80 to 125% bounds. If an effect of food is found in a study like this, the clinical significance of it would be determined based on a drug's exposure response profile. What we would hope to learn from a food effects study is how to administer a drug in clinical trials, as well as how to label instructions on how to take the drug with respect to food, eventually in the drug labeling for patients and healthcare providers. So now I'll touch on hepatic impairment studies and just some brief points about it, such as when one should be performed. If a drug is intended to be chronically administered and is systemically available, if hepatic metabolism or excretion accounts for a substantial portion, which is greater than 20% of the absorbed drug, of the elimination of a parent or active metabolite, if it's a narrow therapeutic index drug, and this is irrespective of the proportion that's metabolized, or the metabolism route is largely unknown. If any of these criteria fit the investigational drug, then the FDA recommends that a hepatic impairment study be conducted. Additional details are listed in the hepatic impairment guidance, which is on the FDA guidance website. Speaking of organ impairment, next we have renal impairment studies. When should one of these be performed? Well, if a drug is likely to be used in renally impaired patients, and when we think that impaired renal function is likely to alter the PK of the drug or its active metabolites, because they are substantially eliminated by the renal route. If it's a therapeutic protein or peptide with a molecular weight less than 69 kilodalton, or drugs that are likely to be used in patients with end-stage renal disease undergoing dialysis, they should be evaluated both while the patient is on dialysis and off dialysis in order to determine if the contribution of the dialytic method to the elimination of the drug and its potentially active metabolites. These details, as well as others, are set forth in the new updated renal impairment guidance that was just put out 2 years ago, or 3 years ago, on the FDA guidance website. So one very important aspect of clinical drug development is the assessment for the potential of a drug interaction with the investigational drug. Now, the investigational drug can be either the drug being impacted, or it can be the drug that's altering the PK of other drugs. So first, we start with in vitro tests or screening to determine if the investigational drug is a substrate for or an inhibitor or inducer of common drug metabolizing enzymes and transporters. These are set forth in the guidances for industry on the FDA guidance website, and we've divided it into 2 guidances, one for in vitro and one for in vivo, and these were just updated in 2020. And some of the more common enzymes or drug metabolizing enzymes are CYP3A4, TC9, PGP is a very common transporter that we recommend screening for as well. If any of these initial screening tests are positive, we recommend conducting an in vivo drug interaction study to confirm the involvement of the drug, whether the drug is a modifier or it's the substrate. Then, based on the results of these drug interaction studies, it can have some wide implications for drug labeling. These implications can range from something informative such as drug X is not a substrate for CYP3A-mediated metabolism, all the way to a contraindication for the use of this drug with other drugs. In order for drug interaction studies to be informative, they need to be designed very carefully. There are some key points that need to be considered when conducting a drug interaction study. Some of these would be factors that need to be taken into account in order to maximize the possibility of detecting an interaction. So keep in mind that we're trying to put forward a worst case scenario, if you will, so that we can find out if there is a true potential for a drug interaction. So we need to select a proper dose for the inhibitor or the inducer. We need to think about the route of administration, the timing of the co-administration of the 2 drugs, as well as the number of doses to be given of each of the 2 drugs. The degree of the effect, either inhibition or induction of a drug metabolizing enzyme, is typically classified by the change in the substrate's AUC. So it could be a weak inhibitor, a moderate, or strong inhibitor. Here, the exposure response profile, again, is important for the drug in order to assess the clinical relevance of any change in AUC caused by either an inhibitor or an inducer of the enzyme of interest. So next, I'll just touch very, very briefly on physiologically based pharmacokinetics, or you may have heard PBPK. PBPK modeling is grounded in what we know to be true of human physiology, and there are a lot of inputs that go into these models. In theory, this approach allows us to make predictions based on our knowledge from previous trials and previous in vitro investigations, and also sometimes preclinical studies. So what exactly is it? PBPK is a mechanistic modeling approach which utilizes all of these inputs, preclinical, in vitro, in vivo data, to help us predict the behavior of drugs in humans. And what we typically apply this to is to explore the effects of various intrinsic and extrinsic factors on human PK. Some of these factors can include, age or ethnicity, the underlying disease status, or even sometimes drug interactions. So another method we use in order to make some predictions of drugs without directly and empirically testing it is model-informed drug development. And I guess in technical terms, PBPK can actually fit under this larger umbrella of MIDD, because MIDD is an approach that involves developing and applying exposure-based biological and statistical models derived from preclinical and clinical data sources to inform drug development or regulatory decision-making. So it's a pretty broad umbrella. And also, having well-timed and well-designed dose-ranging studies or dose-finding studies are important for avoiding dose selection pitfalls later in development, and MIDD can help with these dose selection type of issues. A few years ago, the FDA initiated an MIDD paired meeting program that allows drug sponsors and drug developers to meet with the review team, which is, in the case of MIDD, led by Clinical Pharmacology. Some of the focal points or issues that have been discussed in the past have been dose selection or estimation, clinical trial simulation that's based on drug trial disease models. These may be used to inform the duration of a trial or even selecting appropriate response measures, predicting outcomes, etc. We've also had discussions about predictive or mechanistic safety evaluations. In these cases, we've used systems pharmacology and other mechanistic models for predicting safety, and also identifying critical biomarkers of interest. So we've had a lot of successful cases of MIDD already. And last but not least, I'd like to acknowledge Kelly Reynolds for the use of some of her slides, and Leonard Sachs for the invitation to do this presentation, as always. Thank you.
Well, thank you very much, Paresma, Nikolett, and Shirley, for really outstanding presentations on topics that are fairly impenetrable to clinicians like myself. We have a lot of questions from the audience, so let me get to the first here. Let's see. The first one would be for Paresma. This is a CMC question. If a new formulation is introduced in the clinical trial, what is the expectation in terms of differences between the submission timings of the revised protocol and the CMC amendment? Any thoughts on that, Paresma?
Thank you for that question. I want to start off by just acknowledging that oftentimes we do see changes in the drug product formulation quickly during development. And what we often see is a variety of strategies with submission of that new formulation to support a revised or a new clinical protocol. What we would recommend is that the CMC information supporting a new or a revised protocol, if it's different from the original submission, that information be submitted either before submission of the clinical protocol or along at the same time as the new or revised clinical protocol. One thing that I would not recommend that becomes challenging from our perspective is if a protocol is submitted that proposes use of a new formulation, but the corresponding CMC information has not been submitted to the IND. So this oftentimes leads to us having to request that information from an FDA perspective from the sponsor. So that's kind of just information about the submission timing and strategy. I am also going to ask my colleague Nikolett Biel, who represents Pharmacology, Toxicology, to also provide some additional input from her perspective as well. So thank you.
Thank you, Paresma. So one thing to note from a non-clinical perspective is that typically when a new formulation is introduced in a clinical trial, sponsors often submit a single dose PK study in one species of animals, just to demonstrate that the PK characteristics of both formulations are comparable, so there wouldn't be any exposure differences with the new formulation before administering it to patients. Thank you.
Thanks very much. Thanks, Nikolett and Paresma, for discussing new formulations. Nikolett, I have another one for you. The question is, the purpose of the investigator brochure is to adequately inform investigators participating in trials of a drug. And the individual is asking, what is the FDA looking for when they review the non-clinical portion of the investigator brochure during the 30-day initial IND review and for subsequent amendments to the investigator brochure? Any thoughts about what we look at? And
yes, thank you for that question. That's a very good question. So the investigator's brochure will summarize all the data that the sponsor submits to the IND. We review the original study reports. So, with a 30-day safety review, we're looking at proof of concept studies, whether the sponsor has demonstrated that the drug's mechanism of action is what they claim to be, that there is activity, from an oncology perspective, that they've showed activity, anti-tumor activity, whether in vitro, in vivo, or both. And then, of course, we assess the safety, through safety pharmacology studies and toxicology studies, and whether those studies are adequate to support a clinical start dose. I've touched on this during my presentation, but sometimes when 3-month toxicology studies come in, there might be a new toxicology finding that may not have shown up in the 1-month study, in which case, this would be included in the investigator's brochure amendment. And, we would look for that information to make sure that it's, sufficiently addressed. Thanks again. That was a good question.
Thank you. So I have a question for Shirley. Food can be highly diverse based on its composition and may contain various proportions of carbohydrates, fats, and proteins. How do you account for this in terms of an ADME study? Any thoughts there?
Yes, thank you for that question, Leonard. So we have a food effect guidance which directs the developers of drugs to conduct food effect studies in the worst case scenario, which for most drug products is a high fat meal. So it is the best idea is to conduct a food effects study with a meal that contains very high fat content, and we do specify the different compositions of food that can be used. And for drugs where there may be a concern of exposure with food with low fat contents, you can conduct a study where you compare high fat meals to low fat meal on the effect of the drug exposure. So it just depends on the drug product itself, but yes, definitely the composition of the food or meal can have an effect on a drug product, and that is oftentimes formulation specific. So if a different formulation is developed, we recommend that a new food effect study be conducted with the new formulation. Thank you for that question.
Great. Another question for Paresma. The question is, what is your recommendation on the approach that industry should use to determine and ensure that batch data submitted are sufficient to support regulatory review and approval decisions? So this is the question on batch data.
Thank you for that question, Leonard. So at the time of an initial IND submission, our focus from the CMC perspective is really on safety, and we really focus on the differences between the submitted toxicology and clinical batches, and understanding the differences in, and kind of in the simplest example of of a small molecule drug substance and drug product, really focusing on the differences on the impurity profiles there. And so what we ask is that sponsors really outline the differences between the toxicology batches and the proposed clinical studies, clinical batches, and then provide any justification for why impurities may be greater in the clinical batches compared to the toxicology batches, and information on the levels that have been qualified of those impurities as well. So I think the other piece of this I want to clarify is that we have sponsors take very different strategies associated with submitting batch data. What we really want to see is assuring that the drug substance and the drug product can both be manufactured. And this doesn't necessarily mean that the clinical batches have to be manufactured and submitted. Ideally, that would be the case, but even submission of engineering batches of the drug product may be sufficient to just demonstrate that the product can be manufactured. And we do oftentimes see times where a sponsor may submit both that the toxicology in the clinical batch will just be the same. So they may only submit one batch. So it really depends on the study that's being proposed. I hope that answers the question. Thank you.
Thanks very much. I think that's a really insightful answer. Let's turn to Nikolett again. This one's slightly shift in focus. The question's about an immunotherapy toxicology study. The question is whether that's dose independent. How is the clinical start dose decided on with a fixed dosage? Perhaps you can unravel that for us.
Yes, thank you for that question, Leonard. Again, good questions. So hopefully I understand the question properly. So for immune modulating agents, the start dose is typically determined based on the minimal anticipated biological effect level, or MABEL, which really takes into account all the available data that the sponsor submits, including pharmacology studies, especially that are relevant to the mechanism of action of the drug and potential for cytokine release syndrome, and toxicology studies that are submitted, and any clinical experience that may be available with a similar drug, we can take into account. Sponsors often use PK modeling, taking into account, all the data that I just mentioned when they propose a start dose. And as far as dose escalation with immune modulating agents or biological products in general, following a half log or less increments is generally acceptable. So that's the short answer. I hopefully answered the question. But a lot goes into it. It depends on each product, but really it takes into account all the available non-clinical data to determine a start dose. Thank you again.
We are apologies. We are having some difficulties. Leonard, we had put you on mute because we were getting background noise. Could you go ahead and check to unmute yourself before you proceed? Thank you.
Thanks, Jeff. It looks like we were having a little bit of trouble with audio from Shirley, so I'm looking for another question here for one of the other folks. Nikolett, there's a question which I'm not sure if there's enough information to be able to answer, but the question is, what if the reprotox is not available, if the reproductive toxicology is not available? Any thoughts on that?
Yes, thanks, Leonard, for that question. So often, at the first in human IND stage, reproductive toxicology studies are not available, and that is fine. The potential for embryo-fetal toxicity may be expected based on the mechanism of action of a drug. For example, drugs targeting rapidly dividing cells, we would expect there to be a potential for embryo-fetal toxicity. Often clinical trials will include recommendations for contraception to prevent pregnancy during and for a particular time after the last dose of a drug. And then as drug development proceeds, before a marketing application would come in, the reproductive toxicology studies would be available. And we can use that information to recommend contraception timings for, when the drug is approved to include in the drug label. Thank you.
Thanks, that makes excellent sense. I'm wondering if we've got Shirley back. Shirley, if you're available, again, the question was on in what scenarios would conducting drug interaction studies be appropriate?
Hi. Yes, I thank you for that question, Leonard. I am back. For drug-drug interaction studies, we do often recommend an in vitro study approach first to determine if a drug might be a substrate for or a modifier of certain drug metabolizing enzymes. So we recommend an in vitro screening strategy prior to decisions on whether or not to study the drug in a clinical setting. So if it comes up as a positive in the in vitro screening, we recommend clinical trial investigations and drug interaction studies, as they're called, to further evaluate the magnitude of the effect in in vivo setting. Thank you, Leonard.
Great, thanks for a good answer there. Paresma, one for you. What kinds of CMC issues are noted by FDA during the review of a first-in-human phase 1 IND study? Any thoughts on that?
Thanks for that question, Leonard. I want to say that I very briefly covered this in the presentation, but I'm happy to discuss some of the CMC issues that are often considered safety concerns that may lead to holds or clinical holds. I do want to note by just kind of clarifying that for a phase 1 first-in-human IND submission, the review team is really focused on safety issues that may lead to concerns for CMC of identity, purity, strength, or quality of the investigational drugs, and that's based on our regulatory requirements. So some of the CMC issues that we do see includes not having sufficient characterization of the drug, and in particular, when thinking about drugs that are manufactured using fermentation or recombinant technology, this includes not having sufficient characterization data for the cell banks for manufacturing. One of the other common areas is lack of information on the impurity profiles, and I mentioned this previously, but that comparison between the toxicology and the clinical batches. So if there are impurities in the clinical batches that are not supported by or qualified by the toxicology studies, that may lead to some concerns. The other ones are oftentimes related to the drug product formulations, where the formulations have not been optimized and may lead to things like precipitation of the drug. And this oftentimes leads to concerns that we don't know the strength that's actually being dosed, and the obvious safety concerns associated with the formation of particulate matter in the drug product. And this may be in the drug product itself, if it's in the solution, or when being administered as part of the in-use compatibility studies that are submitted for drug products. So those are just a few of the examples that we do kind of see and want to assure sponsors are looking out for. The other thing I do want to note is it's really important, and we can't emphasize this enough, that sponsors consider meeting with the agency with a pre-IND meeting, particularly for the first-in-human submissions, because I think it really helps to assure that their clinical protocol is also on target, and that the dose strengths that they're going to need for the clinical study have been thought about, as well as any potential hold issues can be identified from both CMC but also other disciplines as well. Thank you.
Excellent. Thanks for a great answer there. Very important stuff to consider. Shirley, one for you. When during drug development would you recommend a food effects study be performed, and why? Thoughts on that?
Great. Thank you for that question, Leonard. So we often recommend food effects studies be completed as soon as feasible. And that is because we need to know whether food will alter the systemic concentration of a drug. And we often need this information prior to additional clinical trials that come down in, phase 2, phase 3, those further downstream clinical trials. It is important to know what food might do to the bioavailability of a drug so that we can help decide what to instruct patients and how they take their medications. So the sooner we have that information, the better, in order to de-risk drug development later on. Thank you.
Thanks very much, Shirley. Here's one for you, Paresma. At what stage during clinical research should the drug product manufacturer be identified?
Thanks for that question, and this is a really good question. The information about the drug product manufacturer, the name and address, is actually a regulatory requirement for IND submissions from the time of opening that IND. And so this is actually part of our CFR regulations. The addition, deletion, or change of any of that manufacturer information after that initial phase 1 or original IND submission should be reported to the agency. What I want to point out here, and maybe where this question is going, is, when do you have to have that final commercial drug product manufacturer identified? And that is kind of a different question. So there's a regulatory requirement that whoever is making that drug product be submitted in the IND, and as changes happen to that manufacturer, that information also be submitted to the IND. However, the final drug product manufacturer that you may be planning to use for a commercial or a marketing application may not be finalized until later in development. Oftentimes, this is happening at around the end of phase 2, and sometimes even around the time of the pivotal clinical studies being run or prior to marketing. So what I would say is, as those later stage changes to drug product and or drug substance manufacturing sites are happening, that sponsors come and meet with the FDA as part of maybe a CMC focused type C meeting to discuss their proposed manufacturing plans as they get closer to marketing. Thank you.
Thanks very much. Thanks, Paresma. Nikolett, another question for you. This is sort of a different kind of different emphasis. What is the rationale for selecting higher order mammals, for example, canine or primate, for IND-enabling safety studies?
Thank you for that question, Leonard. Yes, so, we typically expect a rodent, two species, a rodent and non-rodent toxicology studies to support an IND. The reason being that sometimes higher order mammals, dogs or monkeys, may be more sensitive to certain drugs. It could be a different way that they're metabolizing the drug. And so this could lead to a lower clinical start dose that may be supported by a non-rodent species compared to the rodent species. Also, for biological pharmaceuticals, many times the rodent is not a pharmacologically relevant species, and maybe only the non-human primate may be the only pharmacologically relevant species. And so that would be the species to conduct a toxicology study to support, a clinical trial and assess a safe starting dose in humans. Thank you.
Great, excellent question, and certainly a very comprehensive answer. I've got a question which is addressed both to Shirley and Nikolett. Does the FDA require all the laboratories where these preclinical pharmacology toxicology studies are performed to be good laboratory practice certified, GLP certified? Maybe start with Shirley. What's your thought on that? And then maybe we can move to Nikolett.
Yes, thank you for the question, Leonard. Actually, this question is much more suited for Nikolett, so I'll let her go ahead and take it. Thank you.
Sure. Thank you, Shirley, and thank you for the question. Good question. So typically pharmacology studies, using cells or mice, for example, tumor-bearing mice, they're not expected to be GLP compliant. However, toxicology studies, safety pharmacology studies, genetic toxicology studies, reproductive toxicology studies should be GLP compliant. Oftentimes safety pharmacology studies can be included as part of the toxicology, pivotal toxicology study. Or some sponsors may decide to do separate standalone studies, but they can be rolled into general toxicology studies as well. Thank you.
Thanks very much. I think we're almost out of time. Let's get one more question in. This one is for Paresma. Let's see. It is understood that drug product batches manufactured for clinical trials are typically still going through the improvement process that require adjusting specifications. Should the level of good manufacturing practice for clinical batches be the same with commercial batches? If not, please advise where GMP focus for clinical batches should be. Any thoughts on that, Paresma?
Hi, Leonard. Thank you. That's a really great question. And I will start off by just saying, yes, it is very often that we see significant changes to chemistry, manufacturing, and controls from the original submission of an IND as you move through clinical development. So it doesn't always happen, but it is a very common aspect that is changing during development. As the formulation is kind of being optimized. What the question is really getting to here is about GMP expectations here for phases of development. So what I want to highlight is the regulations for cGMP are actually outlined in 21 CFR 210 and 211. Those regulations are really focused on commercial manufacturing of products and may not be appropriate for phase 1 clinical studies necessarily. But for phase 1 clinical studies, the FDA has actually published a guidance, and I highlighted that guidance on my resources slide. That guidance is specifically titled CGMPs for phase 1 investigational drugs, and those focus areas for INDs in phase 1 are in that guidance, and the requirements include thinking about well-defined and written procedures, adequately controlled equipment and manufacturing environments, and accurately and consistently recorded data from the manufacturer. So what I would say is phase 1 is where the expectations can be slightly different and outlined in the guidance. For phase 2 and 3 manufacturing, those phases of development are subject to the CFR, Parts 210 and 211, and the regulations for commercial manufacturing that are outlined in those parts of the CFR. So thank you.
Great, thanks for an excellent and comprehensive answer. So I guess that brings our session to a close. Again, thanks to our speakers for really an outstanding review of the non-clinical and early clinical studies that we may come across, and for a really excellent review response to the questions. We will now take a half-hour break, and we will return half an hour's time. Millie Dougal will be leading that session. Thank you very much.